Hello, my name is Liz Suter, and I'm an assistant professor of environmental science. Today I'm going to review lesson 4 of Amplicons, which is on preparing output from Amplicon pipelines such as CHIME or Data2, and taking those data and preparing them for statistical tests and plotting. Share my screen. So this lesson will go over importing files into R in case you were working in CHIME and they're not already in R, removing contaminating ASVs, checking sequencing depth by looking at rarefaction curves, removing singletons from your data, normalizing and transforming your data, and then exporting files for later use, either as an R object or otherwise. So the data that I'm using here are the same data that were used in the CHIME and Data2 tutorials in lesson 3 of Amplicons, and they're also the same data that were used in the Happy Belly tutorial that is linked in those lessons as well. So this lesson is available as an R Markdown file, so I'll post it as an R Markdown file and as the HTML file. So the lines of code, if you've been taking the R lessons, you'll recognize this format. The lines of code are in these blocks, these code blocks, and I can run them by essentially pressing play in the corner here. But I'm going to walk through what each of these code blocks is doing. So the first thing is I'm loading a bunch of packages that I used in this tutorial. So Tidyverse is a general like data manipulation and plotting package. And then some of these others are very specific to sequence or ASV data. So PhyloSeq is one with a lot of functions for playing around with Amplicon data. ReadR is something helpful for importing data into R. This is for manipulating sequences. Decontam is a package that we're using for removing contamination. And then APE is also for manipulating data sets, and Vegan is a statistical package that we'll continue to use in lessons later on. So I'm going to run this entire thing, let those packages load. And the output in a markdown file should come right under the block of code. So we'll just give that a chance. And essentially it tells you all of those things loaded. Next, what I'm going to do is take the data that I generated in the CHIME2 pipeline, which is lesson 3A. So I have those data in a folder here called CHIME2 export, and I'm just gonna open that up quick so you can see. This is exactly how the folders looked in lesson 3A once I was done running through that tutorial. So there's a tree file, there's a FASTA file with our sequences of our ASVs, there's a count table, and then there's the taxonomy file. So here I'm importing the count table, which is a TSV, a tab separated value, from that folder. And when I import it, I'm telling it that the row names are there, that the row names are the names. So I'm saying column to row names, so convert that column into names of rows in my table. I'm also importing the taxonomy, which is a TSV file. The tree file, I'm importing with a function called readTree, which is from PhyloSeq. And that is imported in, so that tree file was exported from CHIME as a special format called newic.nwk. And PhyloSeq can recognize those, or this readTree function can recognize those types of files. And then I'm also importing the FASTA file with read.fasta, which is a function from my seq in R package. So I will run this entire code block. And you can see my data files, or all 4 files are now in my environment here. The other thing we need here, or at least later on when we do statistics, is information about the specific samples, like what the sample types are, the environmental metadata, essentially. So that is available from the original Happy Belly tutorial. And I also linked it in a GitHub as part of lesson 3A. So I'm just going to read that directly from the GitHub link. And I also did some manipulation here to delete a row um, in that file. So this file was originally formatted for QIIME 2, and it had a row that indicated what the different columns were, what type of data they were. And here we don't need that, so I just deleted it. So now all of the data are in our environment. The first thing we're going to do is remove contamination. So this is done with a function called decontam, or a package called decontam. And there's a paper about that specific package. And this, I'm essentially replicating exactly how it's done in Happy Belly. So the first step here is to make a logical vector. So logical is just telling for each cell there's either a true or false value. So in this logical vector, I'm saying the first 4 values are true and the next 16 are false, because later I'm going to use this logical vector to indicate that my first 4 columns of data are my blanks. So those are what you are going to be used to indicate contamination, and that the next 16 columns are my samples, so they're false. So I run that. And then the other step that I'm taking here is converting my count table into a data frame. So when I imported it above, the function that I used was a tidyverse function. So it imported my data as a tibble, which is a specific data type used in tidyverse. And that I want it as a data frame because decontam expects it as a data frame. So I use this function called asDataFrame, and I'm calling it count table df. So that just converts it. Then we run the isContaminant function from decontam. So this is going to look for contaminating ASVs and put them in contam df. And one caveat here is that I'm using T to transform my count table to essentially switch the rows and the columns. So when you transform a matrix, you swap the rows for the columns. So the isContaminant function expects a certain format, and my table was in the opposite format, so I just transform it there. And then I'm going to pull the contaminating ASVs into a table and take a look at them in the output down here. So let's run this whole code block. So it's giving me, first of all, it found 13 ASVs that were considered contamination, and 3,401 remained. The second output here is the actual ID of the ASVs. So this is the feature ID from QIIME. This is sort of a number that QIIME associated with the ASV that was called. But here, if I scan over to the second column of data, I can actually look at the taxonomy of these. And you can see that a lot of them are human-associated bacteria, so Burkholderia, Pseudomonas, even Shigella, so things that you would expect to be associated with humans, not necessarily the environmental samples that we're looking at here. So this is page 1, and I believe there's a couple more on the second page. Yeah. So again, more taxa that you would expect to be associated with human contamination. So that's a good sign. We're detecting that. And this is pretty much what they found, or what Mike Lee found in his data set in the Happy Belly tutorial. So next is to take those 13 ASVs and remove them from each of our 4 important files, right? So we have our FASTA file of sequences, we have our count table, we have our taxonomy table, and we have our tree file. So each of these sets is removing those 13 ASVs from each of those files. So I am just going to run this code block. The one additional thing that it's doing is also removing the actual blank samples from the count table. So in the count table, remember my first 4 columns of data are the blanks, and the next 16 are the samples. So now that I've used the blanks to indicate contamination, I can essentially remove them from my data because I'm not going to use them anymore. So I'm removing contaminating ASV rows, and I'm also removing blanks from the blank samples from the count table, those columns. So I'll run all that, and our new data sets appear in our environment here. So you can see my ASV FASTA is now an ASV FASTA no contam because I removed those contaminating ASVs. I also have a count table, no contamination, a taxonomy, no contamination, and a tree, no contamination. So those are my 4 new files that I want to work with. One additional thing I want to say about that code block is that I removed those contaminating ASVs in different ways depending on the file type. So for example, here in most places I'm using like generic R, base R syntax, but in the tree file, remember the tree file is a special format that I imported with filoseek, and base R does not know how to deal with those file types, but filoseek does. And so filoseek also has functions like this one called prune taxa, which can help you remove taxa from a tree file. So that's what I was doing here. I was removing from my tree file those contaminating taxa. So the next step is to take a look at rarefaction curves. The rarefaction is used to take a generic look at the sequencing depth and how many ASVs you have per sample. It used to be used to say something more broadly about diversity of samples, but people are moving away from that because it's very hard to compare from study to study based on rarefaction. But anyway, within a single study or within a single sequencing run, it does give you a sense of which samples were sampled more deeply or maybe not deeply enough, and also how many ASVs there are relative to each other among samples from the same sequencing lane. So here I'm converting my count table without contamination into a data frame again, because I'm going to use a function from vegan, and vegan expects a data frame, not a tibble. Then the function that I'm using from vegan is called rare curve. So as input, I put my data frame count table there with no contamination, and I'm transforming it again because vegan also expects the rows as columns and columns as rows. So the opposite of how I had it before. And then all of these other parameters in here are for customizing the plot. So you'll see that like we have a certain color code from the sample info tab from Happy Belly. So I'll color code the lines and the size of the lines and the size of the points and labels and so on. And then this last line is also adding a vertical line to the plot that will show us the sequencing depth of the least deeply sequenced sample. And you'll see that in the plot when I run this. So they're color coded by sample type. And this is the vertical line I was talking about. So BW1 in this case was the least deeply sequenced sample. And so this is just for essentially comparing across samples. So we can see how many, if we had sampled each of those to the same depth, that lowest sample size, how many ASVs we would have gotten in each of those samples. But samples like these were sequenced even deeper. So the general take home here is that those brown and black samples have more ASVs or more species than the blue and green samples here. So those general trends were also seen in the Happy Belly tutorial, so that's good. We're essentially replicating the same thing. If you look very closely at the rarefaction curve here and the one in Happy Belly, there are some minor differences, and that has to do with the way that we assigned ASVs in the first place. We use different pipelines with slightly different parameters, and also software packages and things like that we might be using different versions, but the general trends are similar, so that's good. So that sort of gives you a first peek at your data set. The next thing you want to do is remove singletons, and we discussed removing singletons in lesson 2 of Amplicons and how this is really important if you have an OTU data set. If you have an ASV data set, it's less important, and especially if you use data 2 to call ASVs. I believe data 2 tries to remove the incidence of singletons before it reports the data. So if you use data 2, you might not have that many singletons, but still we can run this step just to be sure. Or alternatively, if you're looking for very rare taxa, you might want to take a look at what's filtered out. as we discussed in lesson 2. So I chose to showcase 3 different ways of doing this because I thought it was a neat example of showing the difference between tidyverse, base R, and phyloseq. So the first step here is filtering out those singletons using tidyverse. And this is my favorite way of doing it because tidyverse is sort of a really clean, straightforward way. So the function in tidyverse is filter. So essentially I'm filtering my count table based on if any row sum is more than 1. So I want to retain any row that has a sum of more than 1. If it's less than 1, then it will be removed. So that's removing my singletons. And if we run this and we take a look at the output here, we have 3,194 ASVs left from our original 3,401. So it did take out a few singletons, a couple hundred singletons. So that's how to do it in tidyverse. You could also do it in base R. So base R expects a data frame and not a tibble. And here what I'm doing is I'm saying pick out which rows have a sum less than or equal to 1. Then subset that with the brackets and take the ones which are not that, so they're greater than 1, and put them in my new data frame called count table, no contam, no singleton CF. So if I run that, that one also appears, and it's also 3,194, so it's doing the same exact thing. And then the last step is you can also do this in phyloseq. So again, you have to convert the data type here into a phyloseq object, and you do that with a function called OTU table. So I take my count table without contamination, I put it in the function OTU table, and I generate my count table, which is called count table no contam phyloseq. So now this is a phyloseq object, and then I use that with a phyloseq function called prune taxa again. So now I am going to take out any taxa for which the sum, or sorry, I'm going to retain any taxa for which the taxa sum is more than 1. So if it's less than 1, it should be removed. And when I generate that, it actually comes out down here, I believe. Count table. I don't see it here, but if you took a peek at that in phyloseq, it would also have the same number of ASVs. So that's 3 different ways of doing it. I prefer the tidyverse way, again, because I think the functions in tidyverse are more sensible and more human readable, but there's multiple ways of doing it. So the next step here, so we removed our singletons, we removed our contamination. The next step would be a transformation. And I put a bunch of text here based on conversations among Amplicon instructors of different, so there's many different ways of transforming data sets to get it ready for statistics. The first sort of approach at this is what we call normalization. So this is normalizing your your abundance data to 1, or relative to the depth of the least deeply sequenced sample. So if we go back up here, for example, this least deeply sequenced sample was sequenced to about, let's say, 1900. So we could convert all of our samples and resample them to only 1900, or we can take the relative abundance of each ASV within each sample type. That's considered normalization. So for a while, microbial ecologists were using that as a transformation. But we know now that that's not good enough because of the way that Amplicon data are collected. Really depends on how deeply things are sequenced. And so we call that compositional data rather than absolute abundance data. So we don't know that we are sequencing our data sets to their end. So we only consider our data to be compositional and not absolute abundances. For that reason, there's a bunch of different transformations you can do in order to account for that way that microbial ecology data are sampled using next-gen sequencing. And so there's a few references listed here that discuss that and discuss different ways of doing that. There's also a package in Phyloseq which accounts for that called, and so it uses a function called a variance stabilizing transformation that does a transformation that accounts for that. And then the last approach here, which is the one that I'm showcasing here, is called a Hellinger Transformation. And I put the paper for that one as well. And there's some explanation of each of those. So there's no one right way of doing this. It sort of depends on your downstream needs. And you might want to explore this a little bit and see how the different transformations impact your interpretation of your data. But anyway, the Hellinger transformation, which I'm doing here, is built into Vegan. So this decostand function in Vegan is a way to transform data. And I'm telling it, use the method Hellinger. So even the decostand function has different ways of transforming ecological data. So this gets a little confusing because I'm using T again to transform. So there's two different sort of definitions of transform. One is the matrix algebra transform, which literally swaps rows for columns and columns for rows. Or if you have a 3D matrix, it does a whole bunch of other complicated matrix algebra. So that's one definition of transform. And then the other one is actually using some function like the variance stabilization, or taking like the log or the square root, or some more complicated version of that. And that's the other way that I'm using the word transform. So we're transforming our absolute counts into a number that normalizes or accounts for the way that microbial ecology data are collected. So this transformation function also takes as input a count table that is transformed rows switched for columns. So that's what that is doing there. So then I essentially take that output, convert it to a tibble so that I can view it more easily, because I like working in tibbles, although you don't have to do that. And then I just am comparing the first 10 or so, or 10 or so rows here of the transformed and the non-transformed data to take a look. So if I run this, this is what those first few rows look like. So originally I had absolute numbers here, right? So like this ASV had an abundance of 151 in sample R10, but after transformation in R10, it looks something like this. It's a decimal number. So you just want to check how those look, make sure that that transformation is working out. And if you know what your transformation function is doing, like in the case of Hellinger, it's a pretty straightforward, I believe it's, I forget now, but it's like a square root divided by something. So if you know what it's actually doing, you can just calculate it really quickly for a couple of numbers from the original data and make sure that it looks like that in the transformed data set. So our data are cleaned up now, and the last step here is to export them. So again, here I'm doing it in two ways. The first one, I'm taking all 4 cleaned up files, my FASTA file, my count table file, my taxonomy file, and my tree file, and I am exporting them as more generic files so they can be used in any program, like a .fasta and a .tsv and a .nuig. So to do that, for the count table and the taxonomy table, I'm just using write.table, which are sort of generic, either base R or tidyverse functions, or probably base R functions. The write.fasta function is from Seq in R. So in order to work with FASTA files, you need a special package. In this case, I'm using Seq in R package. And then the write.tree is from that ape package that I imported in the beginning. So when I generate this, I'm putting them in my results folder. And if we take a look at the results folder, they're in clean files, tsv, fasta, .nuig. So those are all my clean files. I can come back to R another time and re-import them. Or I can import them into a different type of environment, because FASTA files and TSV files and so on can be viewed as normal text files. And if you have a tree viewer like Figtree or something, you can look at the .NUIC file. The other option is to save all of the files as one object called an R object or an R data object. And you can do this with the save function in R. So I'm saying save my FASTA file, my count table, my taxonomy file, my tree file, as an R data file called clean files. When I run that, it appears as this thing, cleanfiles.rdata. And then later on, if you open a new script or a new markdown file and you want to start working with those, you can just load them all. You can load all 4 files by running this one line, load my cleaned files.rdata. And that's it. So next we'll start working with those cleaned up files in the following lesson of Amplicon.